Moving forward with cells that have had successful recombination of their heavy chain, as mentioned previously, we are at this cell stage of the precursor B cell, where the pre-B cell receptor with the heavy chain and the surrogate light chain are expressed on the surface, and this will signal to the cell for proliferation. And the important thing to remember about this is that one heavy chain in this particular instance, because of this proliferation event, will result in many different B cell receptors, because we have not yet undergone light chain rearrangement. So to explain this in a little more depth, we have our pre-B cell receptors. We have had signaling from our pre-B cell receptors that have resulted in proliferation. So at this point, we have many pre-B cells, all with the same heavy chain, because they all underwent the same recombination events. And so then, after light chain rearrangement of the B cell receptor, we have the potential for B cell receptors that have the same light chain or same heavy chain, but many different light chains. And so this can lead to different antigen specificity from one single heavy chain rearrangement event. And this is referred to as chain pairing diversity, just another mechanism of diversity that's provided during lymphocyte development. So, as alluded to before, when we're moving into the pre-B cell stage, this is where the light chain rearrangement will occur. And something to remember that's unique about the light chain in particular is that the light chain, there are 4 different alleles, 2 kappa alleles and 2 lambda alleles, which allows for 4 chances at recombination instead of the 2 alleles at the heavy chain. So this increases the chances of successful recombination events. And this recombination will also happen sequentially, starting with the light chain, starting with the kappa chain and then moving to the lambda chain. And one noticeable difference looking at the arrangement of these particular loci is they consist of only variable and junction segments, so only V's and J's. They do not have the diversity or D segment that is present in the heavy chain. So there will only be 1 recombination event as opposed to 2. So to give a summary of general path to light chain rearrangement, we have our pre-B cell, and this B cell will increase its RAG expression in preparation for light chain rearrangement. And this light chain rearrangement will originally proceed with the kappa chain at one particular allele. Notice that we are not recombining at both alleles, but we are starting with 1 kappa allele. And if this rearrangement is successful, much like with the heavy chain, we will have allelic exclusion, which results in only 1 light chain being expressed and 1 heavy chain being expressed from the prior recombination event, leading to 1 single B cell receptor for that B cell. But in the event of a recombination event that is unsuccessful, meaning it's out of frame or it does not provide a successful B cell receptor that can be expressed on the surface, then we will move to the other kappa allele. And if this is successful, once again, the cell will be able to move on to subsequent stages of development. And again, if this is unsuccessful, then we can move to the lambda chain to try additional rearrangements, because there are not just 1 light chain, but there are 2 different light chains with 2 different alleles. And if this is successful, the B cell will be able to move on. And you're kind of getting the pattern here, but if this is unsuccessful, then rearrangement will proceed at the 2nd lambda allele. And then if this is successful, once again, the B cell can move on. And at this point, after many different attempts for rearrangement, if this is unsuccessful, this will result in B cell death. So if we have a successful rearrangement and a B cell receptor can be expressed on the cell surface of the B cell, which shows that rearrangement was successful, then we move to the immature B cell stage. But we still have one final test before exiting the bone marrow that is necessary before we have a fully mature B cell. And this process is referred to as negative selection. So we have our immature B cells, which are in the bone marrow, and once they mature, they will be able to exit into the blood and the peripheral lymphoid organs. But this one final check is to make sure that any B cell receptors that are autoreactive or self-reactive, meaning that they can react to host proteins or etc., will be eliminated. And this will be covered further within the context of the autoimmunity lecture that you will have later on in this lecture series. But in the event that this immature B cell, which is in the bone marrow, does not incur an interaction with self-antigen, then it will be allowed to leave the bone marrow. And during this process, it will upregulate this other type of B cell receptor where its constant region is varied, referred to as IgD. And the function of this expression is not well understood, just that it is a way to differentiate between an immature B cell versus that which is mature and is able to leave the bone marrow and enter the periphery. But then, in the event of an immature B cell in the bone marrow that interacts with a self-antigen, in this case a multivalent antigen, which just means that there is more than one of the epitopes that is available for the antigen receptor to bind to, then this results in a strong B cell receptor signal within the context of the bone marrow. And so the B cell will have to modify itself at some point in order to not be self-reactive, or it will simply die. And so the way that it can try and salvage its B cell receptor so that it does not die and is able to exit into the periphery is through this process called receptor editing. So in the event that there are extra light chain segments, not all the alleles had been attempted for recombination, if there are extra segments available for recombination, the B cell receptor will undergo recombination at its light chain once again, trying to form a light chain receptor that when paired with its heavy chain will no longer be autoreactive. So if receptor editing stops the B cell receptor from recognizing self-antigen, then the B cell is salvaged. It will be able to move from this immature stage in the bone marrow to this mature stage in the periphery. There are other types of antigens that can be recognized in the bone marrow, and often these are responded to in different ways. So last time we talked about multivalent antigens, which result in this stronger B cell receptor signaling. But sometimes the B cell receptor will interact with these antigens that are soluble, and this results in a weak B cell receptor signaling pathway. And because the interaction is weaker and leads to a weaker signal, then the B cell will respond in a different way. At this point in time, the B cell will be able to exit into the periphery. However, it will decrease its B cell receptor expression that is IgM. The particular IgM isotype will be decreased, and it is referred to at this point as an anergic B cell, which is not responsive to its self-antigen once it exits into the periphery. So there's not really a risk of it incurring its self-antigen in the periphery at this point because it's not able to respond. It's been changed in some way, and often these anergic cells will end up dying in the periphery, but they are allowed to persist for some time. And then the last byproduct of interacting with self-antigen in the bone marrow is referred to as clonal ignorance. So if there are particular antigens that can weakly interact with the B cell receptor, but they don't actually result in this cross-linking that's necessary for receptor signaling, then the B cell is unaware that it is self-reactive, and it is also unable to respond to these particular antigens. So they're not capable of eliciting this autoimmune response, and they will be allowed to exit into the periphery. Essentially, they are blind to the presence of the self-antigen because they cannot actually signal through their BCR about the presence of this antigen that they're interacting with. So they're not a risk to the host in terms of a potential to stimulate autoimmunity. So at this point, if we have receptors that are not autoreactive, so ones that have undergone negative selection and passed through that particular test, then they move to the mature B cell stage, and they are able to egress from the bone marrow where all of development has been occurring and go into peripheral secondary lymphoid organs. So the way that this occurs is there are specific ligands that are present both in the bone marrow and the blood that either retain the immature B cells within the bone marrow before they're ready to move into the blood, or allow them to exit into the blood. And they are set up in such a way that they have these gradients where in the bone marrow, for example, there's this particular ligand referred to as CXCL12, and it is high in the bone marrow, but it is not present in the blood. Conversely, there is another ligand referred to as S1P that is high in the blood, but low in the bone marrow. So when we have this developing B cell, once it is signaled to move to the mature stage, it is told to upregulate its S1P receptors so it can interact with the S1P that's present in the blood, and to downregulate its CXCR4 so it is no longer retained within the context of the bone marrow. So then at this point, it is able to move into the blood and then into the peripheral secondary lymphoid organs. And the steps of B cell development are not fully over. There are some additional requirements for survival signals to there are additional steps in the periphery in which the B cell will interact with other cellular partners in order to receive these survival signals so that they can be longer lived. And this will happen within the spleen in these particular B cell follicles. So you can see here this follicular dendritic cell, and mature B cell receptors will be able to express these BAFF receptors that interact with this BAFF that is present on the surface of the follicular dendritic cells, and this gives a survival signal to those particular B cells so that they are able to survive. But this is a very competitive environment in which all the B cells are trying to cluster towards the follicular dendritic cells, so the ones that are pushed towards the outside will eventually die, which is another bottleneck in terms of B cell development. And this is the stage at which a lot of these anergic cells are not able to compete with other B cells. And so oftentimes the anergic cells will die at this last stage. And so even though they were able to interact with some self-antigens, they're not able to be competitive enough to receive this BAFF receptor signaling, which is necessary for their survival. So another thing that I just want to mention very briefly is these mature B cells can be subsetted into different cell types. So there are B2 cells, which move to different parts of the particular follicle. Some of them are referred to as marginal zone B cells, which exist on the outside of the follicle within the spleen, and there are follicular B cells, which exist within the follicle close to those follicular dendritic cells. And these are classical B cells, also referred to as B2 cells. But I also want to mention this other less common cell type and contrast it, which are these B1 cells. So first, these B2 cells go through all the developmental stages, which were mentioned previously. And this transitional B cell was the one that needed to receive that BAFF receptor signaling for survival to either become the marginal zone B cells or the follicular B cells. To contrast, the B1 cells, instead of developing within the bone marrow, are produced when humans and mice are at the fetal stage. And all fetal hematopoiesis occurs in the fetal liver. And so these B cells are produced by the fetal liver instead of being produced by the bone marrow during the fetal stage and then persist for long periods of time. And so they go through similar stages of development as the B2 cells, just their origins are different. And another thing that's important about these cells is they have a restricted antibody repertoire, whereas the B2 cells can select any V, D, and J segments. These B1 cells have specific preferences for certain Vs, certain Ds, and certain Js. So just to do a side-by-side comparison of these two cell types, the B2 cells are the ones that are most common, and these are the ones that we have primarily been discussing during the context of this lecture. The B1 cells are from a different origin. They are produced in the fetal liver as opposed to being produced in the bone marrow. And as mentioned, the B2 cells undergo this random VDJ recombination, whereas the B1 cells have specific preferences for certain antigen receptor segments. Lastly, these B2 cells are short-lived and are produced daily, whereas the B1 cells are produced in the fetus, in the fetal liver, and they are long-lived. They move from the fetal liver to other compartments within the body, specifically the peritoneum and pleural cavities, and they usually just stay there and they're self-renewing. We'll get a little bit more into the functional differences between these two cells when we have the subsequent lecture that is on responses, particularly to bacteria. So just to give an overall summary of all the things we've covered about B cell development, we start with our hematopoietic stem cell, which is a progenitor cell that is self-renewing. Then we move to the early pro-B cell stage. This is where the D to J rearrangement of the heavy chain occurs. Then we move to the late pro-B cell stage, where we start with one particular allele and have the V to DJ rearrangement proceed. Then we move to the early pre-B cell stage, where we test for the presence of a functional heavy chain by pairing with the surrogate light chain to try and initiate a B cell receptor signal. And this allows for proliferation of the particular cell, as well as allelic exclusion. So if one particular light chain works, then we turn off rearrangement at the other light chain loci as a way to prevent having multiple light chains. Then we move to the late pre-B cell stage, in which we have light chain rearrangement that will start with the kappa chain and then move to the lambda chain. Then we move to the immature B cell stage, at which point we have...
Did you make a mistake there?
I don't think so, did I?
You were talking about light chain, I think, when you still wanted to be talking about heavy chain.
Sorry, at which point?
Between the late pro-B and early pre-B, and then when you got to the late pre-B, you started talking about light chain rearrangement again.
Oh, wait, yeah, sorry, late pre-B is when light chain rearrangement.
So you said light chain the step before.
Oh, okay, sorry about that. Yeah, I can...
Back up to that slide and I'll match it.
Right here?
Yeah.
Okay.
Hold on 1 second.
Just let me know when I should go.
1 more second.
No worries.
Okay, go ahead.
All right. So after the early pro-B cell stage, in which we have recombination of the D to the J segment, we have recombination of the heavy chain V to DJ segment, which proceeds at one allele first. And then, if unsuccessful, we'll move to the second allele of the heavy chain. After this point, we want to test for the presence of a successful heavy chain. So moving into the pre-B cell stage, we have expression of the heavy chain receptor with the surrogate light chain, which is standing in place of the light chain that is to be rearranged to test for successful rearrangement. And if successful rearrangement occurs, this is where we have proliferation of the pre-B cell as well as allelic exclusion of the other heavy chain. Then, moving to the late pre-B cell stage, this is where we start recombining our light chain, starting with the kappa chain and then moving to the lambda chain. Here we have 4 attempts at recombination with 2 kappa alleles and 2 lambda alleles. And then we move into the immature stage, at which we undergo negative selection. So the B cell needs to see if it is capable of interacting with self-antigens, and if it is and they are soluble, this is often when the B cell will become anergic. Otherwise, it will be eliminated by apoptosis or undergo receptor editing so that it no longer interacts with host antigens. So then we move into this mature B cell stage, at which the B cell will exit the bone marrow and move into the spleen, where it needs this BAFF receptor signaling. So it is a transitional B cell looking for BAFF receptor signaling by interacting with those follicular dendritic cells. And at this point, some of them will move into the marginal zone of the follicle, or others will move to the follicular zone of the follicle, and we will talk about functional differences between marginal zone B cells and follicular B cells later on. But just something to note is that because of the location of the marginal zone B cells, because they're located on the outside of the follicle, they're typically referred to as the first responders of the B cell immune response. So now we are coming to the point at which we conclude the lecture, and I just want to go through the primary objectives, which are the things that we should have learned over the course of this particular lecture. So first, we now know the stages of B cell development that occur in the bone marrow before the B cells mature and move into the periphery, and how they correspond to the specific stages of VDJ recombination. We also learned about allelic exclusion, which is the mechanism by which B cells enforce the expression of a singular B cell receptor on their cell surface before maturation. And lastly, we learned about the mechanisms of B cell negative selection, which are necessary for the B cells to not be autoreactive so that they do not stimulate autoimmunity.